So good morning, good afternoon, and good evening, depending on which part of the world you are. So this is Dr. Pradeep Falke. I'm the Managing Director of Eurofins Biopharma Testing India, and I welcome you all on behalf of Eurofins for this webinar, which is on extractable leachable studies for pharma and medical devices companies. So first of all, thank you all for registering for this webinar and taking out time from your busy schedule and attending this webinar. So before we actually start, I would like to take 2 minutes for all of you to take you through the introduction of Eurofins lab. So I'm going to share my screen and give you some updates about uh, the Eurofins capabilities. So as you can see, Eurofins is a global company with over 950 laboratories across 60 countries with more than 63,000 staff, and it's known as a Eurofins Scientific across the globe, where we are world leaders in food, environment, pharmaceutical, cosmetic product testing, as well as discovery, advanced material, and agroscience testing. So today the webinar is hosted by the Biopharma Product Testing Lab. So I just wanted to, give you a quick brief idea about what this biopharma testing lab across global network. You can see on this map, there are almost 40 Eurofins BPT laboratories across the world with more than, presence in more than 20 countries and more than 7,300 employees across the globe. So last year, June 2024, we have set up a new lab here in India, which is a state-of-the-art lab. So those who joined early could have seen the video which was running, which shows you the state of the lab with all the high-tech instrument as well as a lot of SMEs which have been recruited into the lab, and we serve for all the major customers, domestic as well as global. So in short, just wanted to take you through what are the different services we offered out of Biopharma Product Testing Bangalore, India lab. As you can see, we provide end-to-end services for the pharmaceutical analytical testing, whether it's drug substance, drug product, intermediate, or excipient. The major portfolio where we serve our customers is the method development, method validations, method transfer, nitrosamine analysis, extractable leachable testing, elemental impurities, microbiology lab, genotox testing, stability studies, as well as the characterization studies. So we have all the high-end latest technologies to support your testing needs in all these areas. And our lab is US FDA approved lab, as well as this ISO 17025 approved lab. So what I'm trying to do is now to get into the today's topic, which is on the extractable leachable for pharmaceutical and medical devices companies. So we have invited two distinguished speakers. The first speaker is Dr. Simon Carrera, who is representing our biopharma testing lab in Italy. So he's a business unit manager, and he's also the senior scientific director handling the extractable leachable as well as the packaging testing department in Italy. So Simon has earned his degree in biotechnology from University of Milan in 2005, and he has done extensive work in the biomedical research, developing many methods in the LCMS and developing it for the pharmacokinetic as well as the ADMET profiling. He has led many projects in the areas of analytical chemistry, including the method development, validation by LCMS, MS, GCMS, and drug product impurities characterization. The important point to mention about Simon is almost for the last 10 years, he has focused extensively on extractable leachable testing to support the drug manufacturing companies and addressing the regulatory queries for all his customers. So I welcome Simon for this webinar as a guest speaker. We also have a second speaker who is Udatu Trinath. He is currently the senior manager in the Eurofins Biopharma testing in India. So Trinath also brings extensive analytical chemistry expertise. He holds a master degree in chemistry and has been working with Eurofins since October 23. where he has led many extractable leachable projects, specializing. He's basically specializing in packaging material and medical devices testing. Uh, Almost 7 years, he has exclusively worked in extractable leachable studies, and with total more than 18 years of experience in the characterization, development, and validation of the drug substance and drug products. Like Simon, Srinath has also worked extensively with many global companies and global clients, where he is supporting uh, all those companies in addressing their regulatory queries, and particularly in extractable leachable testing, which is our topic of interest today. So with that introduction, I would hand over it to Simon to take us through his presentation and share his knowledge in this area. Simon, over to you.
Yes, thank Pradeep for this kind introduction, and welcome everybody to this this webinar. I will share my screen, so just a minute. Just let me know if you can see it.
Yes, we can see.
Yes.
Okay, thank you. Okay, so I would like to speak today with you about the USP665 and the USP1665, which are the regulation that were published in the USP in 2020, and these are concerning the items that are used into the manufacturing process. So what is requested is to characterize all of them in terms of compounds that could migrate and could be present therefore into the study protocol, the samples, the sample that we are administered to patients. Before entering into the technical aspects, I would like to share with you some thoughts about how much plastic is surrounding us. You can see it when you go to the supermarket. You see your favorite food and drinks packaging into several types of polymers, and also plastic is widely used into cosmetics. What actually is released from these materials into the product really depends on several aspects. For example, the aggressiveness of the product contained, or the storage condition, on several other factors. Of course, the toxicity and the impact on the body is especially critical when we are talking about the drug products, because mainly because of two reasons. The first one is because of the patient safety. We are delivering drugs to patients that are not healthy. They have some disease, and therefore we need to be very careful to administrate a product that contains foreign substances. The second reason is the drug efficacy. In the moment a compound is released into a drug product, it could have an impact on the potency of this product, especially on biologics or also cell and gene therapy products. Therefore, it is very important to address this assessment, and this is what the authority would like to see in the filing in the dossier. Before starting to talk about what the authority expects to see in terms of compliance with this new regulation, I would like to clarify some point about what is an extractable and what is a leachable. When we talk about extractables, we refer to compound that could migrate from a material to a solution, but we are referring to normal laboratory conditions. So we are simulating what could happen during the real usage. The material in this situation are put in contact with extraction solvents, which are more aggressive with respect to the product itself. But in that situation, we would like to simulate what could migrate, what could happen in real situation after 1 week, 2 weeks, months, or years. When we talk about leachables, we are referring to the same, of course, the same compounds, but the focus here is the product. So in this situation, we test the product, and we do perform the analysis in a real use condition. So I would like just to make a clarification about this wording. Extractables and leachables are present in several parts of the analytical field, and we have to do extractable testing on primary packaging. We have to do that on medical devices, and we will see later with Trinad about the chemical characterization on these. Now, I would like to focus your attention on process components. So USP665 and 1665 deals with all these kind of stuff: filters, tubules, gaskets, whatever is in contact with the product need to be characterized. The most updated regulations and guidances are surely the USP, these two chapters. So my suggestion is always to refer for, at least for now, to the regulation from the USP. In the case of a primary packaging, when we evaluate how we need to conduct the study, the extractable leachable study on a primary packaging, we need, first of all, to define which is the risk dimension. So the risk is defined on toxicological risk and chemical risk. Toxicological risks are related to the route of administration, the daily intake, the duration of the therapy. The chemical risks are related instead to the material, formulation, storage temperature, shelf life. All these parameters enter into the game to define which is the most proper strategy and analytical testing to be in compliance with, to check, to screen for all the potential extractable that could be present, could be transferred for the material into the product. But when we talk about process items, especially a manufacturing line, like could be this one, a CAR T cell therapy manufacturing process, we need to consider that sometimes we are dealing with hundreds of different items: vials, flask, filtration, tubing, gaskets. So in this situation, of course, it is always mandatory to proceed with a risk assessment analysis, because we need to understand which of the items should be treated as a low risk, medium, or a high risk, because we will see later on, depending on the category, we need to address different analytical testing on them. So how can we conduct a proper risk assessment? The steps that are defined into the USP1665 are four currently. Step 1 is the preparation. Step 2 is the risk assessment. Step 3 is the data generation and evaluation. And step 4 is the compliance. Let's go into it for more details. So what means mapping our process? So collect data. It means that we need to collect a huge amount of data which is related to the process flow. So we need a diagram, number of components in the process, total length of the tubing, number of filters, and we need also supplier information and documentation. But it's not enough. We need, of course, to take into consideration the process condition like temperature, duration of the contact, if the process stream is fluid or not, the volume which is in contact with the item, if there are any treatment of the item that could represent a mitigation factor, for example, flushing, sterilization, purification, cleanings, and last but not least, drug product information, dose regime, target population, and route of administration. Of course, these are a high number of information, and this is why this step is usually the most time-consuming of the entire risk assessment. What is in the scope of the risk assessment? What is in the scope of the USP665? In the scope, we can see the pharmaceutical drug product, biopharmaceutical drug substance, and drug product manufacturing components. So this component could be polymeric single-use and multiple-use system. The streams could be liquid or semi-solid, and there's need of a direct contact between the item and our stream. And it is important in the case that the predicate item does not exist. Predicate item is an item, for example, if we are talking about testing a filter, very similar to the one that is under investigation, but we already have information extractable, leachable on these type of items. Of course, there's need to have a lot of similarity between these two items in terms of usage, in terms of composition of materials. But in the case we have performed previously an extractable study on this item, this is the predicate item, I can transfer the same result to a new item if they are close enough in terms of usage, in terms of composition, etc. What is out of the scope? Out of the scope are all those manufacturing components of the APIs. API is not a matter of the USP665. If we have items that are constituted by stainless steel or elastomeric component, it should be out of the scope. Even if we are talking about gaskets, also in this situation, it's very frequently that they are in the scope. If the streams are solid or gaseous, these are out of the scope. And of course, if the items are not directly in contact with the stream, with the product or the ingredients, this is out of the scope as well. And of course, last but not least, if there is a predicate item and I can transfer the information to a predicate item, so a previous analyzed items, if I can transfer those results to the new one, I can, of course, consider it out of the scope. Okay, but in what consists the risk identification? Of course, we have to define two different categories: the likelihood of leaching and the likelihood of persisting of a migrating compound. Likelihood of leaching means that a compound compounds have different propensity to leach, to pass, to migrate into the product. And likelihood of persisting, if the compound is migrated, there could be some purification step, for example, that could eliminate this migrating compound. So the combination between the probability of the leaching power, therefore, and the probability of that compound to persist and to be at the end present into the drug product, these two parameters are those that are the main important to be considered for the risk identification. In terms of analysis, we need therefore to take into consideration the component propensity to leach something, the drug product leaching power, the contact condition driving force, the purification steps, and the drug product related risk. These enter all into the game, into the risk evaluation. And if you put on a matrix the likelihood of leaching and the likelihood of persisting, we can divide our item in three different categories. Those items that belongs to the higher risk category, those that are belonging to the medium risk, and those that are belonging to the low risk. Why the authority are asking to define this category? Because they know that each manufacturing plant, manufacturing line could have hundreds of different items. So it is very important to address properly the analytical testing to each one of them. In the case of a low-risk compound, a low-risk item, the purpose, the analytical purpose is to confirm experimentally the theoretical low-risk outcome. So we test the extraction solution after the contact with the item by non-volatile residue and UV analysis only. And the solvent used is ethanol water 50-50. If after our risk assessment that item is belonging to the medium risk category, we need to generate data to address the risk. And these data are concerning the chromatographic techniques, GCMS, LCMS, and so on. Also in this situation, the solvent is ethanol water solution. The worst case situation is the high risk. Of course, if our item belongs to this category, we need not only to perform the analysis on chromatographic techniques, but we need to add also a technique that is able to screen for the metal release, the potential metal release. And in this situation, the solvents that are used are three. The one is the previous we have seen, ethanol water solution, but also in this situation, we need to add a pH 3 solution and pH 10 solution. These are the solution on which we are going to test the metals, the metal release. Okay, so going forward to our risk assessment, we reached now the situation in risk control. Risk control means that we need to test, to perform the extractable study on our item. So the item need to be pre-treated as it is in a real condition. So if it is sterilized and flushed, then we need to test the item in that situation. We apply the proper solvents, the surface to volume ratio of 6 square centimeter per ml. This is the suggestion of the USP665. Temperature and time of contact is very well defined, 40 degrees, and a number of days of contact that represent the worst case. And of course, type of contact is dynamic when possible, and we need to apply the analytical techniques, analytical evaluation threshold context. So at the end of our risk assessment, we need to find out if we can accept or not the risk. So if we have 10 different items, we have to consider those results, those extractables that are coming from the testing in a cumulative toxicological evaluation, because it could happen that the same compound, the same extractable could be released from the different item of the process. So we are going to have a sort of cumulative effect in the drug product, because the different sources make a sort of additional amount of that specific extractable. So the amount that could reach the patient is higher. In that situation, of course, the source of the extractable and leachable data could be different. It could be a new extractable study. It could be a gap analysis on a preview extractable and leachable study that have been performed, so the client already have in their hands, or could be also the supplier of the items. There could be data coming from them also. So after the gap analysis, if the data are okay, are good, then all these are taken into consideration into the toxicological evaluation. So going now into a more analytical part of this presentation, I would like first of all to show you which is the standard workflow of ENL testing. We have a first step, which is the extractable assessment. A second step is the toxicological evaluation of the results. So the outcome of the extractables study is the input for the toxicological evaluation. And then after the toxicological evaluation, there will be the leachable assessment. So we will focus in that moment, in that step, the attention on those compounds that could effectively, potentially could pose a toxicological concern. And so let's evaluate each one of them. Extractable assessment is what is actually defined, talking about the process material, what is defined in the USP665. So in the case of, if we can summarize the request, we can do that by considering these 5 key points. Item selection, which are the items that we need to test. Extraction solvents, pH 3, pH 10, ethanol water solution, like I previously mentioned. The extraction condition, 40 degrees, but how long? It's enough 7 days. We need to go for 21 days or more. These are defined in our risk assessment. So I will stop a little bit on the analytical techniques that need to be used. In this situation, if you remember the metrics, we have different level of risk, low, moderate, and high. In the case of low risk, the chemical testing is the NVR and the UV absorbance. In the case of moderate risk, we need to add the chromatographic analysis, and we screen for non-volatile compounds by LCMS, semi-volatile compounds by GCMS, and headspace GCMS that is used for solvents or very, very high volatile compounds. In the case of the risk level high, we need to add in the chemical testing also usually the ICP-MS, because MS is usually much more sensitive with respect to optical ICP. Going forward to the last part of this workflow, we have the reporting threshold. What is the reporting threshold? Is the level that is set that allows us to do not report the extractable that are below of it. So it makes a sort of cutoff. So if we have a lot of peaks in our chromatograms, this AET is a level that we can use to cut off the non-toxic compound, because we can understand before the testing which of them are not toxic, because we can set the proper analytical evaluation testing threshold. So let's pass now into the toxicological evaluation. For reason of time, we cannot spend too much word on that, and I'm not a toxicologist, absolutely, but I would like to focus your attention on two specific parameters, the maximum daily intake and the permitted daily exposure. Maximum daily intake is the maximum amount of an extractable that a patient can have daily due to the release from the single or multiple use system. So this is the amount that is coming from the extractable study, the amount that you can see in the table of that study. Permitted daily exposure instead is the daily amount of that extractable that a patient can tolerate per day. So if we put in ratio these two parameters, we obtain what we call margin of safety. In the case the PDE is higher than the MDI, we are on the safe side. In the case the MDI instead is higher, so the maximum, the amount that I found into the extractable study is higher than with the amount that I can tolerate daily, of course, I can have potential toxicological concern. In that situation, the toxicologists have these three different scenarios. The first one is the lucky one. The compound is identified, and there are literature data available. Therefore, the toxicologist can perform the full toxicological evaluation, which is usually the most reliable. If the compound is identified in the second scenario, but the data are not available, the tox data are not available, we can perform what we call read across. So maybe there is some toxicological data which are related to a compound which is very similar in terms of structure with the one that we are looking for, and therefore we can transfer those tox data to our compound as well, or QSAR model that are in silico prediction. The unlucky case is surely the compound, the situation in which the compound is not identified, and of course there could be no literature data available. In this situation, the toxicologists will use the worst case, so the lowest threshold possible. And this is the reason why, when there is an unknown compound in an extractable studies, is quite common that it is also a potential concern, because the toxicologists use a worst case approach. Okay, so going into the end of this presentation, we are reaching to the leachable assessment. So we have concluded the tox assessment. There are few concerns on few compounds, and therefore, if you remember, we are in a situation in which the MDI, so the amount that was found into the extractable study, is higher than the PDE. So in this situation, so the margin of safety is less than 1, and we need to perform, first of all, the identity confirmation, because we have to consider that during the extractable study, not all the compounds are 100% identified. Some of them could be tentatively identified because the information that the analysts get from the mass spectra are not enough to define completely the structure. So In the case of a tentative, we need to purchase that reference standard on the market to inject it into the analytical technique and to confirm the retention time and also the mass spectra. After the identity confirmation, we can perform the analytical method setup and the method validation, because now we are talking about an impurity in a finished product. A leachable is actually that, an impurity in a finished product. So we need to perform the method validation and then to perform the analysis at the end of the manufacturing line, because we are considering the manufacturing line from this moment. We're not considering the packaging, the primary packaging. So if you want to check if the compound is present, it could be better to test it just right at the end of the finish line to the filling machine. So just to conclude, I would like to focus your attention on few take-home messages. First one is that the USP665 compliance need to be reached within May 2026. So there is more or less 1 year, and 3 years have already been passed since the publication of the USP665. So from the authority point of view, there will be no delay, no postponing of this delay, of this deadline. So it's the risk assessment actually is really time consuming, especially the mapping of the process. For big pharma, just thinking about the big pharma, how many different manufacturing line and different plants they have all around the world. Sometimes they could be hundreds, and therefore multiply this manufacturing line for the number of different items they have. Of course, the number of information that they need to collect is huge and take time. It's very important in your risk assessment to do not miss any important information or item, because if you conclude the risk assessment and you realize that some information were wrong or some items were missing, you need to repeat the risk assessment at the beginning, at least considering this new information, losing time. And the last but not least, the analytical condition need to be customized on the real usage of the items. So the design of the experiment and the 5 key points need always to be fully rationalized into the study protocol and in the study report. Otherwise, there will be surely deficiency from the authorities. So I would like to thank you a lot for your attention, and there will be time for answer to question at the end of the presentation from my colleague Trinad. So thanks for attention, and see you soon.
Thank you, Simon. Rinad, over to you.
Hi, hello everyone.
Hi, Rinad.
Just I will share my screen. Hope it is visible for everyone.
Yes.
Yes.
Yeah. Hi, this is Tanan. Today I'm going to deliver how to design a chemical characterization study for medical device in a better way to inline with the regulatory requirements, ISO guidelines, and FDA. Coming to the presentation, everybody knows extractable leachables, how it forms, and as a brief introduction, basically extractables are the estimation of release of chemical substances in a laboratory aggressive conditions, whereas leachables are the measurement of chemical substances under clinical conditions of use of medical devices. Whereas always leachables are subset of extractables, but all the leachables are not extractables. Coming to chemical characterization study. What is chemical characterization? Basically, chemical characterization is like identification and quantification of constituents from medical devices. Then what is the purpose of doing chemical characterization? To identify and quantify the chemicals that is leaching from the medical devices during its clinical use. Why to perform this chemical characterization? To ensure medical devices do not release any harmful chemicals, that is considering patient safety, whereas to in line with the regulatory compliance, to meet regulatory requirements to prove your medical devices are safety to use, and to check the device performance to ensure medical device functions as intended. And coming to the scope, it is applicable for all medical devices. Then, when do I need to perform E&L study or chemical characterization study for the specific device? First, we have to find out what are direct and indirect contact of the medical devices part with patient. If these are identified, then you have to find out what are the materials and chemical composition for the specific direct or indirect contact parts. If any predicate devices are available in the market with the similar or material equivalence, then no need to perform chemical characterization further and risk assessment as per ISO 10993 is sufficient. Whereas if no predicate device is available with similar material equivalence, then we have to establish full quantitative composition of the same. Once after establishing the quantitative composition, then chemical levels which are below toxic concerns and above toxic concerns to be identified. If there is no any above toxic concerns, then no further chemical characterization is required. Risk assessment with ISO 10993 is sufficient. Whereas if any chemical levels which are above toxic levels, then we have to do extra tool study for the specific medical device at clinical exposure to find out what are the materials that are above the toxic concerns. Still, there is no any chemicals which are above toxic concerns. Again, there is no requirement for chemical characterization. Directly, you can proceed for the risk assessment. Whereas, if anything is found above the toxic concerns, then again, leachable study need to be performed. Yet, clinical exposure level. If anything is find above the toxic concerns, then risk assessment for the specific impurities to be done at toxic level. Then, if chemical characterization need to be performed, then what is the process and what is the key steps that involves in the study designing? First is information gathering. What are the devices, components, and materials, like in terms of material of constructions, and what are the compositions for the same? Then what is the manufacturing process that involved to design the medical device? Next, if any processing related sterilization or rinsing process is being involved before its clinical use, Those information need to be gathered to design a better way for the chemical characterization study. Then next it involves test article extraction. Basically this test article extraction will be having some subparts like solvent selection, then extraction conditions. These conditions again like volume selections, extraction volume selections, what temperature and time need to be selected. Again, AET calculations to define some threshold limits to identify which are more toxic and which are less toxic during its release of leachable studies to be evaluated. Then analytical testing requirement, like what are the recommendations as per the regulator requirement for the selection of analytical techniques and surrogate standards and sample preparations. Next step is data reporting. These everything are the steps, key steps that involves in chemical characterization process. Say suppose solvent selection. There are three steps of solvents will be there, which is like polar, semi-polar, and a non-polar. As per the guidelines, like a polar solvents can be selected water or saline solution, whereas semi-polars can be selected as a range of different type of solvents, dimethyl sulfoxide, acetonitrile, methanol, acetone, ethanol, etc. Whereas coming to non-polar solution, there is toluene, cyclohexane, heptane, and N-hexane are the solutions can be selected as a non-polar solvent. It has to be keep in mind that always while selecting these extraction solutions before proceeding for the extraction, there should be a development process should be done to check the compatibility of solvent with respect to medical device to avoid degradation or integrity of the medical device and swelling, any particle generation. To avoid these things, there should be a cross check should be done before initiation of extraction with respect to solvent and medical device. Basing on the outcome from that, proper solvent should be selected. Then extraction volume consideration. Again, extraction volume consideration shall be done basing on basically two type of parameters. One is surface area of medical device, other one is weight. If surface area of the medical device can be measurable, then first choice is to consider the surface area. If the medical device is in a irregular shape, then the second choice of extraction volume selection is basing on weight of medical device. And whenever considering surface area, if thickness of the medical device is less than 0.5, then 6 centimeters square per ml is the ideal case to select the volume while extraction. Generally, examples for this will be like thin films, sheets, and tubing walls. Whereas, if the thickness of medical device is 0.5 to 1 mm, then extraction volume selection will be 3 centimeters square per ml. In this case, examples for the materials will be like tubing wall, slab, small molded items can be considered. Whereas if it is more than 1, not only point 0.1, 0.5 to 1, and if it is more than 1 mm thickness, then those things also can be fall under to select extraction volume of 3 centimeters square per ml. And examples for this is larger molded items. Whereas when surface area is irregular, for which surface area cannot be calculated, those cases an extraction volume can be selected basing on weight of the medical devices. Like 0.2 gram per ml is the ideal case. if the medical device materials are related to powder, pellets, foam, and non-absorbed molded items, etc. Whereas, say suppose if the medical device is irregularly shaped and low density material, like which is very lighter material. For that case, with a lighter material, small weight consumes more area of medical devices. For these cases, to avoid extraction volume concerns, 0.1 gram per ml will be the better selection for extraction volumes. Say suppose these are like examples for this is membranes and textiles. Say suppose if your medical device is observable medical devices, then you have to test in such a way how much volume has been absorbed and or how much volume of extraction solvent it can be absorbed. Like you have to measure before its initiation, and the same volume will be measured and added to the extraction volume during extraction studies. And recommended extraction conditions basing on duration of contact for specific medical devices. Say suppose if your medical device is limited contact, which is less than 24 hours, then exaggerated extraction conditions or relevant worst case conditions of its clinical exposure will be always suggestible. Whereas if it is prolonged contact devices, then exhaustive or exaggerated extractions can be selected to perform extractions. Whereas if your medical device is long-term contact, which is more than 30 days, then again it is exhaustive or exaggerated conditions can be selected. Whereas if your medical device is prolonged and long-term, then out of exhaustive and exaggerated, there is some more conditions are there, in which case you have to select exhaustive and exaggerated. Whereas if your medical device is like a single-use medical devices, which is in contact lesser than 24 hours, so if it is for several days, multiple use of single medical devices will be used during your medication. Then exaggerated conditions is sufficient rather exhaustive condition. And if your medical device is reusable, that also you can consider as a exhaustive considerations for extractions. And then type of solvents depends upon duration of contact. If it is limited, then polar and non-polar solvents is sufficient to consider for extractions. Whereas prolonged also polar and non-polar. If it is a long term in contact, then polar, semipolar, and non-polar. All these solvents are suggestible. And coming to non-volatile residue, this has to be performed for the specific medical devices whenever you are doing exaggerated extractions. Say suppose for prolonged and long term, depends on its clinical use. And while using these extraction conditions, whatever the vessel that you are considering for extraction should be clean, chemically inert, and it should be in a closed container with minimum dead space. These are the required things while selecting the extraction for containers. Then coming to extraction conditions in terms of temperature and time. Basically, there are different type of extraction conditions as per the guidelines, which is 37 degrees plus or minus 24, 37 degrees plus or minus 72 hours, and 50 degrees plus or minus 72 hours, 70 degrees plus or minus 24, 120 plus or minus 1 hour. Say suppose these Extraction conditions to be selected based on duration of the exposure for specific medical device. And you have to select while doing this exaggerated condition, the specific extraction condition should be selected in such a way its temperature and duration should be in a worst case that should exceed the clinical use of specific medical device. So you have to select in such a way these conditions when you are doing extraction. And one more thing is whenever you are doing exhaustive extractions, the same temperature and time conditions has to be followed during your extraction studies also. And coming to threshold limit setting, which is 80, which is very important for any medical device to determine what are the to determine the leachables whether it is below the safety limits or above the safety limits. Basically, AET will be designed basing on the formula, which is dBT into A by BCD into 1 by UF, whereas dBT, it is a dose-based threshold, which is in terms of μg per day. As per the guideline, generally, limited contacts and prolonged contacts, which are in between less than 30 days or less than 1 month. Basically, 120 μg per day is the safety limit, can be considered. Whereas for the long-term exposures, which are more than 1 month to 12 months, 20 mg per day, and 1 year to 10 years, it is 10 mg per day. Whereas if it is a long-term contact, which is for a lifetime more than 10 years, then 1.5 mg per day is the safety limit to be considered. And remaining things what to be considered in this is number of medical device for extractions, whatever we are taking, basing on surface area of the medical device to be considered this number. And whereas extraction volume, as previously we said, depends on the medical device surface area or mass of the medical device. This extraction volume will be considered, and number of medical device exposed per day. That is one of the more parameter that impacts the AET value or threshold limits. And then dilution factors. Once after extraction or during extractions, if any dilutions or concentrations to be performed, then those to be captured in this to calculate the AET values and uncertainty factors. As you know, uncertainty factor is to calculate the response factor of non-targeted impurities. Then analytical testing requirements. Generally, LCMS, GCMS, and HSpace GCMS, ICPMS, or OAS, and IC. These are the high high-end techniques what can be used to determine the chemical characterization study or to analyze the samples during chemical characterization extraction. If you consider a solvent as a polar, then GCMS, HSGCMS, LCMS, and ICPMS and IC, these everything can be done. Whereas semipolars, if the solvent is semipolar, then GCMS, LCMS for SVOC, VOC, and NOC analysis can be done. Whereas if it is non-polar solvent is selected, then GCMS and LCMS for the determination of SVOCs and NVOC analysis. Whereas if it is like a direct medical device extraction, then it is HSGCMS is the right way of choice to consider the testing of samples. Whereas when you are performing NVR test, non-volatile residue, then polar, semi-polar, and non-polar solvents are the solvents which can be considered for NVR test. Next, As per the regulatory requirements, selection of standards. At least 3 surrogate reference standards to be selected for GCMS analysis and 5 surrogate reference standards to be selected for LCMS analysis. And the selected reference standard should be in such a way which covers volatility in a wider range under molecular weight, polarity, solubility, and a different RT range of entire chromatographic region. Like these are the key aspects which you have to be considered while selecting a surrogate standard for specific technique. Then sample preparations. It's always recommended to go with triplicate preparation of samples to minimize the variability between device to device in their configurations or lot to lot variations. And while doing these extraction studies, one key aspect what you have to remember is physical appearance of the extraction solutions after extractions. Before, if you have to be capture all the observations like color changes or increase in any turbidity, particular matter observations, or any integrity or change in medical devices is observed, those everything to be captured during medical device extraction studies. And coming to data reporting, like qualitative or semi-qualitative or quantitative data reporting should be required. Information for device component and materials should be captured. And AIT calculation along with its justification is required. Justification for the reporting threshold and its relevant toxicological assessment, if anything, shall be captured. List of chemical compounds which was observed above the threshold limits for those mass, if any proposed structure or cache number or its identification status like confirmed or confident or tentative shall be captured along with the quantification levels at which level it has been determined. Then extractable amount of the chemical appropriate units, like if you are measuring any specific leachable in terms of μg per ml, then that should be converted into μg per medical device considering medical device clinical use conditions shall be reported. Then appropriate figures like before and after extractions and chromatograms, if any relevant analytical data, everything to be reported. Next, any approach of rationale for the coherent of concerned substances, if anything was observed, those also to be captured as part of reporting. Then coming to the reference section, ISO 1099318, which is for the chemical characterization of medical devices with risk assessment process, and ISO 1099312 for the sample preparation and the reference materials, and the draft FDA guidelines on chemical analysis for biocompatibility assessment of medical devices. Then TTC applications as per ISO TS 21726, ISO 10991 for the evaluation of testing within a risk management, ICH M7 guidelines, and European medical device regulations. These are the things for reference. Thank you all for your time. Over to Anjali.
Anjali, any questions we can take? Maybe one or two questions quickly.
Yes. So if anyone has any questions, please type them in the chat box. I have one here that I am sharing. I think, Simone, this one is for you. Will the requirements of the USP665 be included in the new European guideline ICHQ3E?
Yes, thanks for the question. This is actually a great question because the ICHQ3E is the upcoming new regulation for extractable and leachable studies from the European side. Currently, this is in late. Of course, there's been a huge delay. The draft of this new regulation should have been available for comments 1 year ago, so they are very late. But I'm pretty sure that in this new regulation, there will be a strong alignment on what the USP665 is asking, because of course EMA or ICH and FDA need to be, of course, aligned on the requests in terms of characterization of all the items on the process. So currently, the USP665 is the unique, which is official, but stay tuned because I think that we're going to see something moving also in the European side within May 2026. Yeah.
Great. There's another one related to the guidelines where they want to know, do process items need to be reassessed for ENL testing if there is any change in the material by the supplier?
Yeah. This is an important issue because it is not currently it's not real understandable sometimes is that the supplier are changing some of the material, but this has a big impact on the extractable, because if they change the provider of the plastic of the case, for example, or they change just a few parts, this could have a huge impact in terms of extractable release, because even if it is a tiny piece, this could be constituted by a bad material, and therefore the release from this item could be very high. So in the moment you have concluded your risk assessment and there is a communication for your supplier of that item that is something is changing, that these need to be addressed in the same way of you did in the beginning. So a new or a gap analysis in your risk assessment, or a gap analysis to understand if this change could have an impact on the analytical testing, and if yes, of course we need to compensate with the analysis. Is not a new extractable study, is a comparison study. So we test the old items, the old formulation item and the new formulation item, and we focus the attention on the new extractable only. So it's not a big study, but of course this is very important to be addressed carefully during the documentation phase.
Great. And I think one that follows on here in the chat says, does BPOG guidelines cover all requirements of USP 665?
Yes, that's another great question. BPOG guidance were in force before the USP 665. So in that moment, there were not other than these regulations that were usable to address the standardized condition on ENL testing on process items. When after the USP 665 has been published, of course, these is the new regulation. So some approaches on the BPOGs were not completely aligned with the new regulation of the USP 665. But if you perform the proper risk assessment already on the BPOG analysis, then will fit as well as for the USP 665. But my suggestion is to evaluate what you have in your hands following the BPOG approach and compare it to the new requirement of the USP665. If any gaps will arise, then you need to fill them maybe by documental review or just with some analytical testing for compensation.
Thank you, Simone. Trinad, there's one for you. While doing leachable studies of any MDI, how can we control the matrix issue as like other drug products?
Yeah, it's a good question. Basically, whenever we are performing this leachable study in terms of medical device testing, the control sample will be considered as a extraction solvent, which has to be considered at the initial stage. Whereas in case of drug product, control sample will be in such a way before its storage conditions at initial stage. Whereas medical devices always it's a device that is in contact with the human body. So initial condition will be in such a way whenever there is no in contact for the medical device with body. In this case, you have to consider control sample as a initial stage of extraction solvent. And after extraction with it for the specific medical device will be considered as a sample. Hope I answered your question.
Yes, so yeah. So there's another one here. How to consider DBT value as per contact duration of medical device?
Yeah. DBT value. Here, say suppose DBT is always cumulative contact duration of the medical device during its clinical use. Say suppose if any medical device is in contact with the body per day, like 30 minutes is the duration time which is in contact with the human body for a duration of almost like 2 months or 3 months. Then total, if it is for 2 months, then 60 days into 30 minutes will be calculated as the entire duration. Accordingly, contact will be calculated basing on the guidelines. DBT, if it is false within 24 hours, say suppose 60 into 30 minutes, which is around 30 hours, then it falls under more than 1 day and within the 1 month. So the duration contact time is 2 months, but cumulative falls under limited contact. So DBT will be calculated accordingly.
Great. And then there's one more in the chat here. What is the role of uncertainty factor in AET calculation?
Yeah. Role of uncertainty factor in calculation is, say, suppose whenever we are doing some extractable study or leachable study, all the impurities, whatever we detect, are not only the targeted element, targeted compounds. So there will be a chances of targeted and non-targeted compounds. Whenever you are doing with the targeted quantification, then you can quantify it directly. Whenever you are performing non-targeted compounds, then uncertainty factor is the factor that minimizes the errors in quantification of the non-targeted compounds. So uncertainty factor is to minimize the errors in analytical estimations.
Yeah, if I can make a comment also on analytical evaluation threshold and the uncertainty factor. We have close to publish an article about the continued request from the authority to push higher uncertainty factor. So since during the analytical evaluation threshold calculation, the uncertainty factor is in denominator. So the result is to have a very, very low analytical evaluation threshold. So a very, very low reporting level. A lot of time. So these could be a good thing because a lower reporting threshold allows us to do not miss any potential extractables, so do not have a false negative, but increase a lot the false positive results. So in our research, and it will be published in next month, so it will be in open source, so the article will be completely available. You will see that actually the continue to push down the reporting threshold is not so good in terms of having a lot of false positives. So I think that we need to find out a good balance with the authority to reach a proper uncertainty factor to be used into the analytical evaluation threshold calculation. But so far there's no specific request unless then define the uncertainty factor special for medical devices on a representative database of potential extractables depending on the material. So if you have 100 potential extractable, you perform the response factor, and the relative deviation on those should be considered for the uncertainty evaluation. But and this is what we can say for the moment. But of course, I think we are going to have some new updated in the next years about that, because these have a huge impact on the analytical testing.
So, Simone, there's just one more there where they say is the AET value is different for extractable and leachables? That's the question.
Yeah, it could be actually, because during the extractable step, especially for uncertainty factor, that's where we're talking just right, we are using a worst case situation. It's happened frequently on pharma side, but it's happened also for medical devices as well. But in the extractable step, we are using a very low analytical evaluation threshold because we want to be in a conservative approach. After the toxicological evaluation, the analytical evaluation threshold that we apply into the leachable study will be instead customized on that precise product. So this is what the toxicologists did. They evaluated the results of the extractable in terms of tox assessment, considering the real threshold that need to be applied on that specific product. And therefore, we use the same tox threshold the toxicologists used in their evaluation also into the leachable study. So this is why the AT from extractable study and the leachable study could be different.
I think that's it for now. Pradeep, do you have something to say?
No, I think thanks, Simon, and thanks, Srinath, for taking us through this nice presentation. As I can see, there are a lot of good comments coming from the attendees. That is very valuable information and very good presentation in this hot topic. And we appreciate your time. And equally, I appreciate all the participants and the attendees of this webinar. So what we want to do is, we cannot take all the questions today, but definitely we will note down these questions and we will try to answer each and every question separately. As you can see on this slide, you see if you have any testing request, if you have any query, if you want to contact us, please use this email ID, which is flashed on this screen right now. And we will be happy to serve you. And I think, Anjali, we can stop this webinar here. Again, thank you all. Thank you, Trinath, and thank you, Simon.